All right, we're ready.
All right. Everyone, we still have some folks coming on in, but we'll get some of the logistics done as folks kind of join us. Thank you all for joining us tonight for HMSC's Virtual Science on Tap. I know that we're not at the Rogue together in Newport enjoying good food and drink, but I hope you, wherever you're at, are comfortable and have something good to eat while we watch tonight's presentation. My name is Cinnamon Moffett, and I'm the Research Program Manager at Oregon State University's Hatfield Marine Science Center, located in Newport, Oregon, and I will be your host for tonight's talk. Just a few logistics as we get started. Wanted to remind you all that this is a webinar format, so a little different than a Zoom meeting. In a webinar format, you do not have control over your mics, cameras, or screen shares, but we do hope that you put any questions you might have for us into the chat box, and I will read those out to tonight's speaker at the end of her presentation, and we will get through all of your questions to the best of our ability. Feel free to put those into the chat at any time so that you don't forget them at the end. I also wanted to just remind everybody that we are recording tonight's event. So if you miss it or you need to see something again, I just put into the chat the link where this recording will be posted, probably by Friday afternoon is my goal. So feel free to share that link with others that weren't able to be here today. A couple of quick little announcements before we get started. Wanted to just do a little quick promo for next month's Science on Tap. That will be February 15th, when Sue Spinagle from the Department of Integrative Biology at Oregon State University at Hatfield Marine Science Center will be giving a talk entitled Peering into the World of Larval Fishes, Tools, and Tails. So I hope you join us for that one on February 15th. If you need any information about our events, login details, start times, any of those kind of things, if you go to HMSC's web page, scroll down to the bottom, you will get a calendar of events there, and you can get all the login information that you might need. But we are excited tonight to have Dr. Sylvia Yamada here to talk with us about her work with green crabs. But first, just a little bit of background on our speaker. Sylvia Yamada is a marine ecologist and faculty member with the Department of Integrative Biology at Oregon State University. She received her bachelor's and her master's from the University of British Columbia and her PhD from the University of Oregon. Her research includes salmon stock identification, predator-prey interactions, invertebrate fisheries, and the role of introduced species in marine environments. She has been monitoring green crabs in Oregon estuaries now for over 20 years. So we are so excited for Dr. Yamada to be here tonight to talk to us about the work that she's been doing. So Sylvia, the floor is yours. Take it away.
Well, thank you very much, Cinnamon, for inviting me. I'm glad to be here. And I want to draw your attention to my co-worker, Sean Schooler from South Slough National Estuary Research Reserve. And also, there we go. Also, many of our collaborators in Oregon, Washington, British Columbia, and at Woods Hole. Now today, I might be able to answer some of the questions that you have about green crabs. Number one, how can you tell them from native crabs? Where did they come from? Why should we care? What's their life cycle? How are they spreading? Can we reduce their numbers? And finally, can we eat them? Now, as the name implies, green crabs are native to Europe and North Africa, but they're not always green. So color is not a good diagnostic feature. There are many native crabs that are green, and green crabs are not always green, depending on since the last time they molted. When they first molt, they're nice olive green. However, as the time between molts increases, they will turn, the bellies will turn yellow, then orange, and then finally red. If you find a crab that's red, it means that crab has not molted in a long time, possibly longer than a year. Now, green crabs are a fan-shaped crab that don't get very big, only about 4 inches from tip to tip on the carapace. And the best way to tell them from our native crabs is to locate their eyes. And then between the eyes, there are 3 bumps. And behind the eyes, there are 5 distinct triangular teeth. Now, Dungeness crabs are also fan-shaped crabs. However, if you count the teeth on the Dungeness, there are 10 very small teeth versus 5 distinct triangular teeth. Furthermore, if you look at their claws, for crabs of similar size, Dungeness crabs have much more delicate claws than green crabs. Green crabs have claws of different sizes. Usually the right claw is larger and it's called the crusher, and the smaller claw is called the cutter. So you might want to look at green crab claws as being a multi-tool. Now, comparing a Dungeness claw to a green crab crusher claw for crabs of similar weight, you can see that the green crab crusher claw is a lot stronger. And we have shown that they're able to crush larger seed oysters than the young Dungeness. Another native crab is the red rock crab, and it can be distinguished by the black tips on the claws, and it has 9 scallop-shaped teeth behind the eyes. Our native shore crabs, the Oregon shore crab and the purple shore crab, they are not triangular, but they are square-shaped and they're much smaller. And if you look behind the eyes, they have 3 teeth. Now, why should we care about green crabs taking hold on our shores? Well, green crabs are predators and ecological engineers. The role of predators has been well documented whenever green crabs are abundant, be it in their native habitat or in some of the introduced places where they are now thriving. When green crabs are abundant, they can really decimate cockles, mussels, urchins, and worms. And the best example of what green crabs might be able to do is the soft-shelled clam industry on the east coast of North America during the 1950s. During that time, there was a warm period, just like now, and the green crabs were thriving. And consequently, the landings of soft-shelled crabs, soft-shelled clams, that they decreased from 14 million to only 2 million pounds. And green crab predation was one of the reasons why that decline occurred. Now, the role of green crabs as ecological engineers has only recently been appreciated. Green crabs have the ability to transform a very productive meadow, eelgrass meadow like this one, to bare substrate through their foraging activities. What they'll do is they'll use their claws and other appendages to sift through the sediment to get at clams and worms and other prey organisms. And in the process, they tear up the plants. Now, eelgrass beds are very similar to coral reefs or tropical rainforests in that they provide structure for many other organisms to find shelter and food. So if you remove them, you can remove a whole community. Now, the life history of green crabs is very similar to that of other crabs. They attain sexual maturity when they're about 35 millimeters or less than an inch and a half in carapace width. Here you can see this female here. It has a yellow belly. And this male has a triangular shaped abdomen, and it's orange. Now, green crabs mate when water temperatures get above about 60 degrees Fahrenheit, and after the female has molted. They can only mate when the female is soft. So females typically will mate, and then they'll store the sperm till about the fall or so, and then they'll exude their eggs. And they're very good mothers in that they fan their eggs to aerate them, and they clean algae off of them. And when the eggs are about to hatch, they turn into first zoea larvae. And these zoea are really cute little larvae. They have big eyes and they have a spine because as a deterrent against predators, because zoea and megalope larvae are prime food for fishes. They're very desirable. Now, the little zoea will swim to the surface of the estuary and ride an outgoing tide to the near shore, where it will rear. It will undergo vertical migrations, feed on the plankton, go through several molts until it becomes a megalopa larva. This is a transitional larva between the planktonic larval stage and the more sedentary adult stage. So this megalopa can swim in the plankton, and it also has legs for crawling on the substrate. Now, what the megalope do is they swim to the surface and then ride an incoming tide back into the estuary, where they will settle on the bottom and metamorphose into young crabs. So in summary, green crabs have a two-phase life cycle. The adult phase is sedentary, lives on the mud flat on the surface of the sea and intertidal, and they can be moved inadvertently along with fishing gear or seafood products that are being transported, and they're wrapped in algae to keep them moist and so on. However, on their own, they can only travel maybe 10 miles or so in their lifetime. However, the larval stage, which is rearing in the open ocean, can get caught in very strong ocean currents and can get transported hundreds of miles from their source of origin. So how do they get around? Well, here are some of the mechanisms. Green crabs spread around the world. In the olden days, like over 200 years ago, they appear on the East Coast on a species list. And most likely they were introduced in sailing ships along with rock ballast. Now, many ports have places called ballast point, where the ships would dump their ballast rocks along with encrusting organisms such as barnacles and algae, and there would be a lot of hitchhikers also attached. So that's how they got to our East Coast, and most likely also to Australia during the gold rush. Now, the green crabs that are living in Cape Town Harbor, most likely they arrived on a fouled vessel called a exploratory oil drilling platform. They move around the world, and some of the fouling from that vessel must have gotten into Cape Town Harbor, where green crabs are now well established. Nowadays, green crabs get transported in airplanes. You can get bait worms from Maine. You can order them. You can order Atlantic lobsters also from the East Coast. And typically, these organisms are wrapped in algae, ascophyllum here, to keep them moist. And what a lot of people do is they take the seafood product and then they throw the algae into the ocean. And sure enough, they were dumped into San Francisco Bay, along with a number of other species, some snails and some young green crabs. And that's how green crabs got established on our coast in San Francisco Bay. Now, once a species is established in a new place, it's got the larval stage that allows them to get transported many, many miles by ocean currents. Now, this slide gives the distribution of green crabs in their native habitat and also in introduced places. And you can see there are two isotherms, or lines of equal temperature, that define the distribution of green crabs, the thermal distribution of green crabs. Now, green crabs have to have temperatures above 10 degrees Celsius or 50 degrees Fahrenheit for at least part of the year in order to grow and for the larvae to develop. So the most northern places you find them is northern Norway and southern Iceland. Likewise, there is a temperature isotherm at 18 degrees in the wintertime, that defines their southern distribution in Morocco and Mauritania. Now, if temperatures never drop below 18 degrees Celsius or 64 degrees Fahrenheit, the females cannot successfully incubate their eggs. And here you see the Mediterranean variety of green crabs, which was introduced to Japan. The first introduction, of course, were over 200 years ago to New England. Also in the 1800s, late 1800s, were introduced into Australia. More recently, about the 1980s, they were introduced into South Africa. And on our coast, they were introduced into San Francisco Bay from the East Coast, and most likely with bait worms from Maine. That was apparently the mechanism. Most recently, maybe 15 years ago, they now also appear in Argentina. Note that green crabs are a temperate species. There were reports of green crabs being found in Brazil and Madagascar, Sri Lanka, Burma, and Hawaii, but none of those tropical sightings took. Now, it's it's over 100 years ago that the famous Norwegian fisheries biologist Johan Hjort made the observation that a lot of North Atlantic species in the North Sea, the fishes landings, they would fluctuate from year to year. And he made the observation that it was the early life history of these fishes, the larval history of these fishes, that experienced the ocean condition that they experienced would set the catches a few years later. So if ocean conditions were warm one year, the larvae would grow well and survive well, and a few years later, you'd have good catches of cod or herring. However, in a year when the currents were unfavorable and took the fishes away from their typical feeding grounds, you'd have a whole cohort of missing fishes. So very similar to what's also happening to green crabs on our coast. So green crabs, the range expansion of green crabs and the abundance of green crabs is linked to ocean conditions when the green crabs were larvae. And it's warm temperature, are good for green crabs. They need 60 degrees for mating, and they need 50 degrees Fahrenheit for larval development, and they need favorable currents. Now, in winter, you have the Davidson current that dominates our coast. It originates in Southern California. It hugs the shore. It flows along California, Oregon, Washington, British Columbia. And it's a warm current, and it can be very strong, up to 50 kilometers per day or 30 miles per day. And as we mentioned before, green crab were introduced into San Francisco Bay during the 1980s or something like that. The populations build up in some of the poorly flushing sloughs, and then when larvae were abundant enough, they got caught in the Davidson current and transported northward. Here we go, San Francisco Bay. The first time we hear of green crabs is in 89, but of course the green crabs were well established by then. 93 in Bodega Harbor, 95 in Humboldt Bay, 97 in Coos Bay, and then in Willapa Bay, Washington, and Vancouver Island in Barkley Sound in 98. Now, the Davidson current can be especially warm and strong during El Niño years. And we had two very recent very strong El Niño. The 97-98 El Niño was the strongest of the last century, and the 2015-16 El Niño was also very strong. And during the 97-98 El Niño, during the winter, water temperatures off Newport were 12 degrees Celsius, well above what's needed for green crab larvae to survive, or 54 Fahrenheit. And the currents lasted a long time, from September of 97 to April of 98. So it's almost 8 months that the currents were favorable for northward transport of water masses and larvae. And velocities were 50 kilometers per day for all of January, February, March, so 30 miles per day. Now let's do the math. In a non-El Nino year, current velocity is only about 6 miles per day. Let's say the larvae are in the plankton for about a couple of months, so they could be transported, over 300 miles from San Francisco Bay to Humboldt Bay by Crescent City. However, in an El Nino year, it's 5 times that, like 30 miles per day. And so they could be transported all the way from San Francisco Bay to Vancouver Island, no problem. So, so after the 97, 98 El Nino green crabs arrive in Barkley Sound, and it's only a year or so until they make it to Nootka Sound, because of the Davidson Current. Also, there is Vancouver Island Current that hugs the shore year-round, and it flows north. So a few years later, they're at the northern end of Vancouver Island. About 5 years later, they're on the central British Columbia coast, and 10 years later, they're on Haida Gwaii, or what's formerly known as the Queen Charlotte Islands. Now, the abundance of green crabs in Oregon and Washington is also linked to ocean conditions, temperature, and currents. So in 1998, the spring and summer of 1998, we had young green crabs in all the estuaries that we examined. So there was a coast-wide colonization that I call it the El Nino cohort, or year class. And in the winter break, I got some students together and we tagged a bunch of green crabs at the Hatfield Marine Science Center. We used spaghetti tags. We inserted them into the gill chamber between the upper and lower carapace. And that seam between those two carapace plates is the first to open up during molting, and the crabs can just sort of crawl out, just like taking off an old pair of gloves. And the tag is retained. Now, green crabs, when they molt, they increase their carapace width by about 30%. And you can see that the larger crabs that we tagged had molted once by July 99, whereas the smaller ones molted twice. Here you can see the growth of the 98 year class. They grow very rapidly in their first year, and by the first winter, they attain 47 millimeter carapace width on average. The second year, about 70 millimeter carapace width, and then their growth slows down, and until age 6, when they die of old age. So in the summer of 2004, I took some crabs into the lab for a feeding experiment, and these crabs were very sluggish and they died for no apparent reason. So it does appear that 6 years is the length of their lifespan, which is also the same in Maine and in Europe. So now, if you have a lifespan of 6 years and you didn't have any more El Nino events bringing larvae to Oregon, you would predict that green crabs would go extinct in Oregon. And they almost did. Okay, here we're looking at recruitment of young green crabs. So these are just the young green crabs that we're trapping at the end of their first growing season on the mud flat. And we trap them in crayfish or minnow traps in the high intertidal in September. When they're in the summer, we can't really trap them because a lot of them are still too small to enter traps. So it's a compromise. So we're not getting all the little guys, but in September, we get most of the New Year class that we come in. And you can see that the 1998 year class is pretty strong in all the estuaries. We average about 100 crabs per 100 traps, so it's 1 per trap. And we don't really get a lot more crabs after that until we get into the next El Nino of 2015-16. Between those two El Ninos, recruitment is sort of hit or miss. We have some years where recruitment is fairly good, and then we have some years where you have absolute recruitment failure. All right. Now we're superimposing winter water temperature off Newport onto that same graph, and our critical temperature was 10 degrees. Below that, they don't develop. And sure enough, 2008, 2012, we don't get any recruitment. And recruitment does follow the temperature curve pretty well up until you get to the next El Nino. So here you can see there's a period of warm winters here, 2003 to 2006, and we got some recruitment. 2010 was a mini El Nino, and then 2015-16 was an El Nino. And after that, we've been getting good recruitment every year, even though, 2020 we shouldn't have because it was cold. We're thinking that we are getting green crab larvae from sources other than just California. So we have multiple sources of green crab larvae now that the populations are larger in Oregon, Washington, and British Columbia. Here we're looking at catches of adult green crabs. So not just the young of the year, but you can see that, there's not much to write home about. Green crabs were very, very rare until we get to 2016, 17, 18, all the way to 21. So the populations are building up because they live for 6 years, and we've had good recruitment in all those years. And what we're seeing is that green crabs, in some places, they average more than 10 per trap per day. And that is considered sort of the ballpark limit at which, above which you might be able to detect ecological impact. So I just want to mention again that now we seem to have multiple larval sources. We get them from California, from self-perpetuating populations in the Davidson Current. Also, Alan Shanks showed us that we can get local reproduction. 1 year in January, February, and March, he was picking up first instar zoea in Coos Bay. So they had to be locally produced. And Carolyn Tepal tells us that some of the samples of young green crabs that we sent her from Washington and Oregon actually originated from a unique population, a genetically distinct population in Souk Basin on Vancouver Island. So those crabs must have arrived as larvae during the summertime when the currents were flowing in the opposite direction. They were flowing from the north to the south. Looking at green crab abundances, in Oregon, green crabs, at the most, we get 20 per trap. Very seldom do we get more than that. At Pipestem Inlet in British Columbia, in Bolinas Lagoon, we've been getting a maximum of maybe 200 per trap. So it's an order of magnitude higher. Tasmania, East Coast, much higher. And in the lagoon in Portugal, you're getting over 100 per trap per hour. So our problem in Oregon isn't all that great at this point. However, it could become much, much worse. So what are we going to do? What can be done? The goals are, the ultimate goal is eradication. Get rid of all the green crabs on the whole West Coast. That may not be possible with a species with planktonic larvae and with multiple larval sources. it's very unlikely that eradication coastwide is feasible, unless we get, like 6 really, really good cold years where they can't reproduce. But, like we did in 2011, 12, and 13. But I don't think that's going to happen. So we need to have smaller goals. Maybe just reduce the impact on our native species, on eelgrass beds, on valuable shellfish beds, and biological reserves. So we have to define what we want to protect, and how much we want to reduce the population. We want to reduce it below probably 10 per trap per hour, or trap per day. What can be done? Well, they can be trapped. That is probably the #1 way you can do it. Also, we want to keep native ecosystems healthy. So we don't want to overfish, like the predators or their competitors. For example, the red rock crab in very saline, cooler, lower estuary will eat green crabs that are the same size and smaller than them. And green crabs would avoid these larger crabs. So we want to make sure that we don't overfish them. Poison bait's been tried, fences have been tried, and people have looked into introducing green crab parasites, but none of these are options. They're just bad ideas. In the future, we might be able to use pheromones to control the behavior of green crabs, such as sex pheromones to attract female green crabs. That would be ideal if we had male sex pheromone that would attract the females. That would be great. And also maybe deterrents, like something that smells bad that green crabs will avoid. So we could protect maybe valuable shellfish beds. Okay, the only place that I know of close by where green crab control has been tried or is being tried right now is in the Washington Salish Sea. The crab team, which is supported by Washington Sea Grant, consists of biologists from the University of Washington, various state and federal agencies, and a whole slew of volunteer trappers. And what they do is each year they monitor green crabs all over the Washington Salish Sea. And when they find green crabs in great abundances, they do an intensive trapping project. Now, as you can see, green crabs right now are not found in Hood Canal, and they didn't find any in the San Juan Islands. So that's a good thing. However, they find them by Bellingham, Drayton Harbor here by the border, by Blaine, and by Bellingham in the Lummi Nation Sea Pond. They're very, very abundant. They've removed 70,000 green crabs last field season, and they're gearing up to do more trapping next field season. So I'll talk about that briefly, and I'll also talk about Dungeness Spit. Now, Dungeness Spit was one of the first places to receive green crabs from the open coast during the last El Niño, when the water in the Strait of Juan de Fuca was warm enough for open coast larvae to be transported to Dungeness Spit. And what the Wildlife Refuge personnel did, they did some intense trapping in 2017, 18, and 19. All through April through October, they set 50 traps 4 times a week. And they didn't remove all that many crabs, only 41 females and 55 males the first year, and so on. However, they prevented this population from establishing itself. The last couple of years, trapping was not as intense. And this last year, they didn't find any young of the year, so they didn't find any recruitment. So it shows that intense trapping, when the population is still very low, could be very successful in preventing a population from establishing itself. Next, we look at the sea pond at Lummi Nation. Bellingham is over here. This is Bellingham Bay. And as I mentioned before, there's a whole lot of green crabs in the sea pond. This is a dike that was built around it to enclose salmon and shellfish so that production would increase. But green crabs got in there and they're using it like an incubator. The larvae are retained and will just keep perpetuating the population. So the goal is for them is they're going to need a huge crew next year to do some intensive trapping in that sea pond. So if you're looking for a volunteer job for the summer, I'm sure they could use your help. Now, I mentioned that native red rock crabs can keep green crabs out of the lower estuary. And here you can see the work by Chris Hunt. He found that red rock crabs will eat green crabs that are the same size or smaller than themselves, whereas they will not eat their own species at the same rate. And you can see here that in Toledo, in Port of Toledo, it's not favorable for green crabs or red rock crabs. But in the upper estuary, where it's warm and less saline, green crabs are thriving. But they are not coming to South Beach Marina, where the red rock crabs are abundant. And some of that could be attributed to the predatory activity of the red rock crabs. Now, larger crabs like red rock crabs would eat smaller green crabs. But we should not neglect our native species, Hemigrapsis oregonensis and Hemigrapsis nudus. These two species have a cousin in Asia, the Asian shore crab called Hemigrapsis sanguineus. And Hemigrapsis sanguineus has been introduced to the East Coast. And people notice that in places, like in very structured habitat, like riprap and so on, and boulder fields, that the green crabs were going down in numbers, and they were wondering why. And they found that Hemigrapsis sanguineus was eating the young green crabs. So all crabs have to be small at one time, and so they're a prey to these Hemigrapsis crabs. And so our Hemigrapsis crabs could have a very similar function on our coast, keeping young green crabs from becoming abundant. Now next, the final one is pheromones. Pheromones have been very successfully used in controlling insect pests. However, they have not been that successful in the marine environment. Pheromones are chemical messengers very similar to perfume that animals use to communicate amongst themselves and between species. And the female sex pheromone, which is simply a molting pheromone, because when females molt, just prior to molting, they'll release a molting pheromone in their urine. And that scent is extremely attractive to male crabs. If you coat a golf ball in female pheromone or molting pheromone, these silly crabs will mate with it. I mean, the scent overrides any visual or tactile cues. So what we did was we received some pheromone from our colleague in the United Kingdom, and we set up some traps. These are folding traps that have a slit-like opening that allows crabs to enter this way and presumably not to exit. And we set those every 10 paces along the shore at Sally's Bend. And some of them would be baited with pheromone in a jelly matrix, and some of them would be baited with placebo or just no scent at all. So here we have a no scent treatment, and here we have the pheromones treatment. And you can see we trapped a whole lot more crabs with pheromone than we did with no scent. And we trapped just as many, if not a few more crabs with a pheromone than we did with tuna bait. So pheromones do hold some promise, but not in the immediate future. There's a lot more research that needs to be done before we can have a practical application in controlling green crabs. Now we get to the final topic. Can you eat green crabs? Yes, you can. They appear on a species list from the London fish market in 1850. So people were selling them then. And also they are harvested in some lagoons in Portugal, presumably to supply the paella market in Spain. And also in Venice, they have a century-old fishery that goes back, 300 years, where people focus on two products. They look at soft-shelled crabs in the spring and crab roe in the fall. So here you see these fishers here. They have these baskets. What they do is they trap green crabs, and then they're very good at telling whether a green crab is about to molt or not. And they'll keep the ones that are about to molt and put them in these baskets and watch them very carefully. The rest they let go. Any crab that molts is removed immediately, and the freshly molted crabs are sold in the seafood market as moleche, and they sell for about $25 a pound. Here you see them being prepared, almost mouth-watering good. And also the caviar or the roe is extracted from female crabs in the fall. Now, if you're interested in harvesting green crabs, you might want to invest in this green crab cookbook, or you might just simply go on greencrab.org and get 20 of the recipes that are described in the book. The question is, should we trap and eat green crabs? And the answer is yes and no. In California and Oregon, where green crabs are everywhere now, the answer is yes. In California, they're classified as a general species, and you can collect 35 per day per person. In Oregon right now, they're classified as miscellaneous species, and you can get 10 per day per person. But the proposals in the works that by spring, you can get as many as 35. Now, Washington and British Columbia, this story is different. It's illegal to possess green crabs. They don't encourage you to collect them. They tell you just to take a photo if you find them in a place where they have not been recorded before, and send it to the respective agency. For example, green crabs are not found in Hood Canal or the San Juan Islands right now. And in British Columbia, they're not found on most of the west coast of Vancouver Island. So here you can see there are some objections to catching green crabs for food. I've been told by a colleague in Newfoundland, where green crabs are extremely abundant, that they will not make a dent. Taking them for food will not make a dent into their numbers. The numbers there are so huge. I mean, in a couple of hours, you can fill up a trap. And they haven't been there all that long. They've only been there for maybe 15 years, whereas in Oregon, they've been here for 25 years. So, and also selling green crab bodies for fertilizer also makes no sense, because it takes too much energy to transport these green crabs. So they have not found a use for them, although they have tried, and they found that trying to get soft-shelled crabs is just a real trick. It's just really hard to do. they've kept them in tanks, and in no time at all, whenever a green crab molts, the other ones will eat them. So we just don't have the skill in telling telling crabs that are just about to molt like they do in Venice. So on our coast, it has not been that successful. And also there's concern about moving green crabs from non-native habitats to uninfected habitats. That's not a good idea. Also, green crabs in Oregon are not monitored for toxins or harmful algal harmful algal blooms like the Dungeness crab is from the open coast. And so we have to be kind of careful. And also green crabs can live in polluted water, so that's not safe for shellfish culture. But if you want to give it a try, some of my colleagues say green crabs taste quite well, and they make pretty good broth for some of the dishes that you might want to have, or like in paella or in bouillabaisse. Go ahead, get a fishing license for $10, but be sure to discard the viscera or the mustard, or what they call the brain, the digestive gland, hepatopancreas, guts and gills, because it's in these viscera that the harmful algal toxin would be most concentrated. So this is all I have to say. And if you have questions, be sure to type them into the chat, and I'll be happy to answer them. Thank you.
Thank you so much, Sylvia. Thank you for sharing and telling us kind of the back history of what we've been seeing. I'll just let you know that Mary says that the Gwynn Library at Hatfield does have a copy of the Green Crab Cookbook. So if anyone's interested, you're welcome to come into the library and check that out. As folks are putting in their questions into the chat, I actually had a clarifying question for you, Sylvia, and I'm not sure how easy it is for you to go back to a particular slide, but maybe you can just tell me. You had a slide that had the number of animals per trap per day, and I was just wondering what year that was. Back a little bit more. What year was that?
Well, that was done a few years ago. And but Yaquina and Coos Bay, they're more recent. Pipe stem Inlet, yeah. So they're not up to date, but I think they would be in the same order. Like our crabs in Oregon are not as abundant as in some other places.
Okay, so
as
relative to the others,
or British Columbia. Mm-hmm.
Yeah, I was just interested because you had said that the 10 animals per trap per day was kind of an important cutoff. And so I was seeing that Oregon here on this particular list had 20. Yes.
Mm-hmm. Well, it's really hard. It's just, you have to look at it as order of magnitude, more or less, like 10 is the same as 20, for example. It's kind of hard, but 200 is different from 20.
Yeah. Okay. Fair enough. We are.
And 100 per trap is different from 20 per trap a day. So,
most definitely.
I mean that trap number per trap is a really rough estimate because it can vary from day to day. It can vary on the type of bait you're using and so on.
Okay. Thank you for that clarification. So I'm going to try to start working through some of these questions, which we're definitely getting. So the first one that I have for you is the state of Oregon or any state agency addressing green crab invasion?
Is any state?
Any state agency actively addressing Oregon, the green crab?
No, I think Oregon Department of Fish and Wildlife, they're watching green crabs closely, but they haven't decided to do anything yet. It's only in the Salish Sea where there's a concerted effort to keep the breeding population low. So what they're doing is they're looking at places where the flushing rate is not very great, or high retention areas like bays and lagoons, and they're trying to remove as many crabs as possible in those places. So they want to reduce the the larval supply in the inland sea. There have been experiments in Pipestem Inlet They have been doing some trapping. Bodega Bay, they have been doing some trapping. And there's a lagoon within Bolinas Lagoon, just north of San Francisco, called Seadrift Lagoon, where intensive trapping has been taking place also. And what people find is that, yes, intensive trapping will reduce numbers, but you have to keep doing it and doing it. And in some places like Bolinas, Seadrift Lagoon within Bolinas Lagoon, where green crabs are the only species, you can have counterintuitive results. Now, it's like a big tide pool, I would say, and green crabs are the only species. And if you remove all the large males over time, the smaller crabs are actually thriving. So you can get actually more crabs by removing the larger crabs. So you want to make sure that you leave a certain number of large crabs in the population to control the younger ones, because they're cannibalistic, yes.
Interesting. It's a complex array of responses to what we're seeing.
Right.
Sounds like. A little bit related to this, can you talk about the cons to raising the catch limit to 35 crabs per day here in Oregon? Do you know the rationale behind that? And I know
well, they want to give people an opportunity to harvest green crabs. Just like they're harvesting red rock crabs, right? They can harvest those up to 35, and green crabs certainly can stand some fishing pressure.
Do you know the cons to raising it to 35? What is the why would we not want to do that?
Oh yeah, they're being raised to 35. That's the proposal.
Okay, I think the question is asking why would we not want to do that?
Yeah, they're being raised to that's the goal, is to raise it to 35.
Okay. Okay, so the next question here is, have you ever eaten a green crab, and what does it taste like? So they want personal experience, Sylvia.
I have to admit, it's kind of hard to eat the animal that you're studying, and I have not eaten it, but I have a couple of colleagues who have tried green crabs, and they say it's quite good. Yeah, it's like any other crab. However, they're smaller crab, and to get at the meat, you have to do a lot of work. For example, there's this recipe for green crab risotto, okay? You need 20 crabs to make the broth, which is doable. But then you need another pound of crab meat, for this on top of that. And where are you going to get a pound of green crab meat? It's going to be a lot of work.
Nice. And do you know the stance in either Oregon, Washington, British Columbia, or even California on euthanizing the crabs when you catch them?
I don't think they have any regulations. Like what? Do you have regulations for Dungeness crabs?
Yeah. Okay.
I mean, it's a food, right? And I don't think they tell you what to do.
Okay. If you have your collection permit.
If you have your collection permit, yeah, you can collect them. And I personally would freeze them first, so I wouldn't be putting them in boiling hot water. But that's what people do. They put the live animals in boiling hot water. And in Venice, what they do is, they have these soft-shelled green crabs, and what they do is they make an egg batter and let them wallow in the egg batter for a couple hours, and then they sprinkle a little flour on them and put them in hot oil and deep fry them.
We got a couple questions here, Sylvia, about Inslee, Governor Inslee in Washington's emergency order addressing green crabs at Lummi. Do you know anything about that proclamation today?
I'm not the best person to talk about that, but I do know that they're getting some money from various sources to pay for the green crab removal. Yeah, it's quite an operation. Yeah.
Yeah. Okay. This might be a little bit hard to answer, but this individual is asking, in those areas where eradication efforts are underway, do we know the the total area of the distribution of crabs? You had talked about like Dungeness Spit was kind of a smaller population.
Right.
I think this question has to do with like, what is the geographic extent of those
right,
areas?
Right. We could easily get that, you know, from a map. Yeah, I cannot give you exact numbers. Yeah.
All right. I see in the chat that somebody actually put in Governor Inslee's proclamation today. So if anybody wants to see the language to that, there's a link in the chat now that you can go ahead and take a look.
Thank you very much. That's very thoughtful.
We are getting lots of questions. I'm going to try to keep up with them. I will honestly tell you if I miss somebody or I don't ask your question. Sylvia, you had up your contact on one of your slides. Do you want to put that back up for us while we kind of navigate through these few more questions?
There we go.
Okay, so this question is about the warming ocean temperatures. Will that help green crabs permanently establish in Oregon's coastal waters?
Yes, I believe so. Warmer water temperatures are very good for green crabs, yes.
So can you elaborate on what research has been done on the testing for domoic acid? And I'm assuming, Ichang, that you were, or Minda, if you asked this question, in green crabs specifically?
Domoic acid?
Domoic acid testing in green crabs. Has there been work done?
Nobody's doing it. We don't know anything about it. Yeah, so right. I can't tell you that.
Okay, and then so this particular question is asking about what the benefits of reducing or eradicating the species from the West Coast would be. Can you talk a little bit more? You had talked about when we get to a certain level of population density, we start to see impact to the environment. Can you talk a little bit more about what we've seen in Oregon, if we've seen that issue here specifically?
I don't think we have seen any ecological effects in Oregon. However, on the west coast of Vancouver Island, they do believe that green crabs are affecting eelgrass beds, and that would be very much of concern because eelgrass are such productive habitats and provide so much shelter, you know, to fishes and waterfowl, shrimp, and other organisms. Yeah, so they do, you know. Shellfish growers had some problems with green crabs when they first arrived in California, but shellfish growers know how to handle crab predation. They grow shellfish in bags, in mesh bags, and they also grow them off the seafloor in bags and stuff like that, suspended bags and in trays. So the shellfish growers are not, as far as I know, are not particularly worried because they know how to handle it. It
probably changes the amount of effort they have to put into raising their shellfish, though.
Right. They have to sort their baskets, whatever. More not let the green crabs get too big. Yeah, that's what they need to do. Yeah.
We have a question here about, can you talk a little bit more about the impact of green crabs on Dungeness and other native species here? Right.
Young Dungeness and green crabs live in the same habitat in the estuary, and in some places, you know, I get Dungeness and green crabs in the same traps, and there definitely would be a competitor for food with Dungeness. If green crabs were to become more abundant, they could eat smaller Dungeness crabs, you know, because they have more powerful claws, and especially any molting crab would be prey, you know, would be prey for green crabs. Also, I think it has been shown that green crabs are better at finding shelter than young Dungeness crab, and they'll displace young Dungeness crab from shelter habitats. That could be a factor. So yeah, they're a competitor and a predator. So that's what we need to consider.
Okay, and this is another clarifying question, just to get a little bit more detail. We've talked about the impact of green crabs on the eelgrass bed. Can you be specific about how they might reduce or impact? What is the behavior that they're doing that would impact those eelgrass beds?
Well, green crabs, they will sift through the sediment. They'll use their claws and their appendages to just sift through the sediment and look for prey. And in that process, they're ripping up the eelgrass plants and destroying them. They also eat the seeds of the eelgrass.
Well, that I didn't know.
Yeah, and they'll eat eelgrass as well. Yeah, but basically they're after shellfish. That's their favorite food.
Nice. We have reached our 7 o'clock time limit. So Sylvia, I just want to say thank you so much for being here and sharing this information with us. It's a very timely topic. So it's just really great to learn a little bit more about what's happening here in Oregon. Do you have any last comments that you want to share with us?
No, anybody that has any further question, I'll be sure to contact me by email and I'll get back to you.
Great. And we do have a few other folks that have put questions into the chat. And so if I didn't get to them again, please reach out to Sylvia. Her email is up on the screen and you can ask those questions specifically. And for everybody online, thank you so much for being here. I know there's lots of things that you can do on your evening. So thanks for joining us on a science presentation. And I hope maybe we'll see you next month for our next Science on Tap. And again, Sylvia, thank you so much for being here. We appreciate your time, your energy, and your experience.
It's been a pleasure. Thank you.
All right, everybody. I'm going to end the presentation now. Thank you, Sylvia. Thank you, everyone online. We'll see you next month. Bye now.
Bye.